Yes, no? Can you wave if you hear me? Okay, great. Before we get into content type things, my secretary asked me to ask you if you have a preference about how the handouts should be put on Icon. So the two alternatives we have are to put them on as PowerPoints or as PDFs. The PowerPoints are obviously a lot bigger, but they also have more versatility in some other ways. So what I'm going to do is I'm going to ask you if you want it put on in a particular format, and if that one is good for you, raise your hand, and you can raise your hand both times if you want to. Okay? How many like having the handouts put on Icon as PowerPoints? Okay, hands down. How many like having them put on Icon as PDFs? All right. I'll let her know that probably we should just do both then. She'll try to mark them as to which is which so that you don't have to randomly download stuff hoping to find one. Are there other course-related questions that you have in the meantime? I'll just make a reminder again that we posted a copy that looks like this, except with very pretty colors, because AJ put some time into putting pretty colors on it, of when you can find one of them or me available to answer questions and so on. Again, I know that the first couple of chapters were more review than the later ones will be, but it's not too early to get started on developing the, your mastery of the material. I also want to just mention briefly the slideshow that I started this session with and the slideshow from Friday. I'll occasionally have slideshows of images that I think complement or talking about. They're not required in any sense, but I will post them on Icon so that if you would like to have access to them for yourself, they'll be there. So any questions on what we covered on Friday about nucleotides and the structure of DNA or RNA? So the pedestrian bridge on Burlington, the Burlington Street Bridge, is that a right-handed or a left-handed helix? Okay. We can follow it later. I just want to touch on what we closed off with at the end of the class on Friday, which is looking at the triplet code. That is, we need to account for 20 amino acids. We have 4 bases to do it. And so we take 3 at a time, non-overlapping, 3 at a time, then move on to the next 3. And the consequence of that is that we have a code that has several very valuable properties. It is unambiguous. If I give you an RNA sequence, you know what the sequence of the translated protein is. It is a redundant code. We can say the word alanine in multiple different ways. So you can look here, and here you can see a cluster of 4 different codons. They all mean alanine, so it's a redundant code. But if I give you that codon, you know it means alanine, so it's unambiguous. But the other is that it is a resilient code, and this was something that was not expected or predicted. That is, given our code, which is shown over here, a mutation is very likely to leave the protein sequence unchanged. It will either be a synonym, so here these 4 all are synonyms, so mutating from one to the other will give you the same thing, or they'll be conservative, that one hydrophilic polar thing might be substituted another one. That was not a necessary property of the genetic code. And in testing for it, only 1 in a million Monte Carlo runs produced a genetic code that was as resilient as the one that we, in fact, owe our existence to. Are people comfortable with the concept of resilience in thinking about a code? I'll be including this particular article on Icon if you want to read it in more detail later on. Okay. So, part of what makes thinking about DNA and its relationship to proteins a little bit tricky is that there's a lot of different kinds of things going on. And in thinking about how we read out that genetic code, we need to take into account all the different kinds of steps that are going on along the way. And what exactly those relationships are, to some extent, varies with where you are in the scale of organization, if you like, the complexity of the organization. So while it is completely true that all protein is encoded in DNA, it is not true that all DNA codes for protein. And in organisms like us and in organisms like sequoias, most of the DNA does not code for proteins, which is what came as, I will say, a great surprise when it was discovered. And to get a sense of just how graphic that is, here is a pie chart for you. If we take all of the human DNA, a good 3/4 of it is never even transcribed. It's there, but it's not transcribed. It's not copied into RNA molecules. Of the regions of DNA that is transcribed, most of that transcript is not translated. And this can take a couple of different forms. Something that textbooks tend not to dwell on is that there are many transcripts in the nucleus that never get out of the nucleus. We don't quite understand what's going on there yet, why the nucleus would bother to transcribe things and then throw them away, but it is a fact. But even for the things that are going to have a product to get out into the cytoplasm, it's not just a quick, there it's go out. And this diagram is meant to evoke for you some of what's going on in that processing and selection process. So if we look at a stretch of DNA, and this one is just taking, for a start, a quarter of a megabase, so 250,000 base pairs here. Let's say that that whole thing is transcribed, and now you have a big messenger, a big precursor to a messenger RNA. And so transcription starts off and creates the RNA molecule. But in complicated organisms, in higher organisms, loosely speaking, a lot of that sequence should not be there in the mature messenger RNA. It has to be spliced out. And so in the process of splicing out the stuff that shouldn't be there in the final thing, you might go from 250,000 base pairs to a mere 6,000 bases in the spliced messenger RNA. So you have, as you can see, comparable to this circle, this pie chart here, you've actually thrown away the great majority of the RNA that was in the original transcript. Now, of this spliced messenger RNA, not all of it will go into protein. There will be an untranslated region at one end and an untranslated region at the other end. And because RNA, like DNA, is a polar molecule, we can speak about the 5' untranslated region and the 3' untranslated region. So if in this 6,000 base messenger RNA, maybe only 4,400 bases are used as codons. And so this one has 1,480 codons, triplets that can code for amino acids. And so now when this messenger RNA is translated, it produces a protein of 1,480 amino acids. So the original stored information in the DNA was 250,000 base pairs. And after all the splicing and pairing away, we're down to a mere 1,480 amino acids. So this is quite unusual. And we'll see along the way some of the reasons why this is a useful thing, but certainly there's still a lot of rather appalling mystery associated with it. One of the aspects about the central dogma that was not appreciated when it was first formulated is that we can call that central dogma recursive. That is, it feeds back into itself. And what I've done is to take the original diagram about the central dogma, which looks at information flow. So the information to make DNA comes from DNA. The information in DNA is used to make RNA. And the information in RNA is used to make protein through adapters, through the transfer RNAs that can match up codons and amino acids. Now, in life as we live it today, when you make DNA from DNA, you also need RNA for that process, and you also need protein. When you make RNA from a DNA molecule, you also need protein and RNA to do that. And when you make protein from an RNA, you need RNA and protein to do that. So this is something where, as processes, they're all entangled together. They're dependent on the product of the process. So that makes life a little more complicated. The other thing to keep in mind is that our DNA is not just kind of a bowl of spaghetti, but it has organization to it in terms of being more and less actively used and in how the different regions within the chromosome are organized. And so it's been appreciated for the last number of decades that when we look at the chromosomes, the compacted DNA that's spread out at mitosis, we can actually talk about inheriting chromosomes or regions of chromosomes, and that process is called cytogenetics, the genetics of the structure of the chromosomes. And cytogenetics can actually let us map the location of genes for where they are, and it can allow us to understand the physical basis for genetic rearrangements, why this particular organism has a different phenotype if it's actually moved its genes around. And this is just showing you a couple of examples of that. There have been, across the years, a number of different methods for detecting cytogenetics. In the earlier method, there was a matter of staining for more and less densely active regions of the chromosome, and you could see things like this where you would have differences in intensity of staining. Now, as I mentioned, we've now gotten to a point where we have chromosome paints, where we can literally paint the entire chromosome one color and its neighbor some other color, and that allows us to refine the results of cytogenetics in greater detail. And this is just giving us a couple of examples of that. This would be using one of the older methods. It's just sort of a darker, lighter method. But here you can see that this pair and this pair of chromosomes, you might have suggested that there's a change there, but it's really hard to tell. With the chromosome paints, you can now see that the tip of this one is yellow and the tip of this one is turquoise. And so there's been a translocation that's exchanged those two regions with each other. Here's an example of a chromosomal makeup that has quite extensive rearrangements. Here's one where most of the chromosomes look normal, but a piece of this one has been transferred over there. Cytogenetic mapping is sufficiently refined. So this is chromosome 7 on a human, and we know, based on extensive genetic analysis, that in this region here is where the gene for cystic fibrosis is found. So I think this kind of brings home the level of resolution that's now possible in genetic analysis. One of the other reasons for being interested in cytogenetics is that as cells progress further and further down the pathway that leads to cancer, somewhere along that pathway at different times for different tumors, they usually start losing control of correct division of the chromosomes for mitosis, and they start to acquire either whole chromosomes or rearrangements. And so chromosomal abnormalities tend to accumulate and become worse and worse the worse a cancer is. So being able to do cytogenetics can be informative in diagnosing and making predictions for cancer. Another thing that we can do now that was impossible just not that long ago is sequencing DNA. When I was a graduate student, one of the members in my class actually got a thesis for sequencing about 600 bases of DNA. It was that difficult at that time. And she had to make an explanation for why she wasn't sequencing the coding region. So, it's gotten to the point now where you can send off samples of DNA and get back the information. But the tricks by which it's done are worth paying a little bit of attention to. So let's say we start off with a piece of double-stranded DNA. And we have, this is the strand that we want to sequence. We're going to add several things to that. We're going to add a primer. And a primer is a short sequence of nucleotides that's complementary to the strand. So in this case, this is our primer. We're going to add an enzyme, DNA polymerase, that can make more DNA. We're going to add deoxynucleotide triphosphates. So we want the right monomers to make more DNA. And then fluorescently labeled dideoxynucleotide triphosphates. So these are nucleotides that have two places in the ribose ring that are deoxy, and they have a fluorescent dye attached to them. So we're going to add all that stuff to this mixture. And as the primer extends, most of the time, it will simply add the correct complementary base pair and the reactions paid for by the triphosphate tail of the nucleotide. But once in a while, it's going to, instead of inserting a normal nucleotide triphosphate, it will insert a 2', 3' dideoxynucleotide triphosphate. And this is, obviously, it cannot add any more nucleotides at the 3' position. Since there's no oxygen here at the 3' position, the DNA polymerase cannot add another base there. It's going to terminate the reaction at the spot where that base was inserted. And remember, this is not just a dideoxynucleotide, it's also then had a dye added to it to make it fluorescent. And so the outcome of it is that we're going to have a whole population of different lengths, depending on what particular position the polymerase randomly added this dideoxynucleotide. And you literally can have gels. In some cases, they now use chromatography, but this is not unrealistic, where each band here represents a separate length of product that ended at that spot because there was dideoxynucleotide inserted at that spot. So sometimes it'll just keep running and running and running and get almost all the way done before it inserts one of the red bases. In other cases, the third base, it happens to insert one of the dideoxynucleotide. And so sequencing this is really just as simple as going along and seeing which base is at which position, because they've been given different colors. Now, DNA, there's a lot of DNA in an organism. This method can handle hundreds of nucleotides. It can't handle millions of nucleotides. And so one of the things that has to be done is to start with the starting DNA and then break that up in one way or another. Sometimes it's done just randomly by mechanical shear. Sometimes it's done with targeted enzymes. And then each of these stretches or segments will be sequenced. And because there's some overlap among them, you can then compare them and add them up. Sort of like, you've got a map that gets to Chicago, but you want to get to New York. So you go to get to Chicago, and the new map you have picks up at Chicago, but gets you to, Cleveland. This kind of overlapping information allows a final reconstruction of the gene sequence. It's not at all uncommon for it to be sequenced altogether 6 different times. So in other words, this piece and then another 4 pieces before they're confident that that particular base is actually G or A or whatever. Any questions? Okay, let's keep moving. Other things that we can do with DNA is we can actually go in and cut specific sequences, which is a little bit amazing if you think about it. Our cells each have this much DNA in them. This is, roughly speaking, a meter of DNA. And each of our cells has that much DNA in every cell. And yet, through using restriction enzymes, we are capable of targeting where we're going to break that up. And this is just giving you an example of a couple of kinds of cleavages. In some cases, the restriction enzyme will make what are called overhanging ends. And so you can see here there's a free sort of flapping in the breeze end. In other cases, they might give a blunt cut like this. And having generated a specific cut in it, we now can combine it with other things. We can make recombinant DNA. So if we start off with a plasmid and some other kind of DNA that we want to insert into it, when we cut both of those the same way, some fraction of them will reanneal. Remember, we talked about melting and reannealing. Will reanneal with the foreign DNA now as part of the plasmid. Plasmid is a small circular DNA derived from bacteriophage or from bacteria or some other sources. And then this is still not yet molecules covalently, but a separate enzyme can ligate that and seal that, so you now have a double helical circular DNA that is all one piece covalently, but now includes the foreign DNA that you want to work with. This whole process has gotten very sophisticated. This is giving you just an example of one kind of cloning vector. A vector is a piece of DNA, or in some cases an organism, that you use to carry something around. And this is one of the widely used ones, PUC18, plasmid, P for plasmid, UC for University of California. And this was the 18th one that they developed. And this particular cloning vector has a couple of markers in it. This here is AMP-R, is short for ampicillin resistance, which means that the plasmid, as in the attack state, has in it resistance to an antibiotic. So only the cells that contain a plasmid will live in the presence of the antibiotic. You can throw away all the cells that don't have DNA. It also has, inside of a gene, multiple restriction sites. So this broad arrow here encodes a protein called LAC-Z, which, when it's intact, can make a colony blue. And inside of this has, in this particular one, has 13 different restriction sites. So you would have a choice of 13 different possible sequences that you could slap into there. And if you insert something into there, you can then disrupt it. And so that allows you to tell that this is not just something that has a plasmid, but it has a plasmid that contains something. Another technique whose power is really still being appreciated is something called PCR, for polymerase chain reaction. Polymerase chain reaction is an example of the power of exponential growth, when you multiply the products at one level by the next ones. Population growth is exponential. That's why dandelions can take over a lawn. Nuclear explosions are exponential. One of the reasons they're so dramatic. PCR is not as much of a nuisance as dandelions, but it can be pretty dramatic. Let's say we start off with a piece of DNA that we want to have more of for a variety of reasons. In order to amplify this target sequence, we need to have separate primers, one for each strand. And you can see little arrowheads if you look in your textbook. I think it'll show up sort of on the screen, to show that they have an orientation to them. So we're putting in primers to the separate strands. And we can separate them by heating them. So we allow those to bind to the strands, and then we use an enzyme to extend from those primers, and now we have two pieces of DNA, one that's been extended this way down to there, this one's been extended up to that way. If we repeat, so now we started with one, now we've got two. If we repeat this again, here we go again, same primers again, same extension process, and each round we're going to have twice as many. So one round is 2, then 4, then 8, then 16, then 32, then 64. A useful rule of thumb, and this is true for anything that doubles, is that 10 cycles is roughly 1,000-fold amplification. So 20 cycles is roughly 1,000,000-fold amplification. So you can start off with a piece of DNA and have 1,000,000 copies of it at the end of 20 cycles. And these are not terribly long cycles. We're talking in terms of minutes here. You might notice that the product of this amplification are pieces that only lie between the primers, ultimately. There'll be a few left that have the ends from the original one, but they're going to especially be enriched in the things that are between the two primers. I think the next slide, yeah. So PCR, polymerase chain reaction, is extremely useful for a number of different processes. Something called site-directed mutagenesis, where it's like a magic wand. You know the site that you're interested in. You know what you think a mutation would do. Wouldn't it be nice to have a mutation there? Well, with PCR, it's possible to target a mutation to the exact base you want to have mutated, if that's what you want, or to go across an entire region or whatever. And to do that, you take advantage of having a slight mismatch in the primers. You remember I said that perfect complementarity gives the best binding in the highest melting temperature, but a partial similarity allows you to have annealing under more permissive conditions. Well, you would pick conditions here that are a bit more permissive, and you could use primers that are different at that site, and then the product is going to have the mutation that you've inserted in there. PCR is sufficiently sensitive that you can find pathogens that are 1 to 10 per mil of blood. So these would obviously not be bacteria you'd worry about at that point. These would be the things like parasites, where most of the parasites like to live somewhere else. But if you get into the bloodstream, PCR can detect them at the level of 1 to a few per mil, which otherwise would be impossible. PCR allows the police to amplify even tiny amounts of DNA found at a crime scene, so that if the criminal has shed a hair, if the criminal has left a little bit of saliva or enough pieces of skin, the amount of material needed to detect DNA using PCR is remarkably little. Now, I mentioned that our DNA is this long. We've got about 1 meter in each cell. Surely they don't sequence the whole 3 billion base pairs to do criminal identification, and they don't. What they take advantage of is the fact that when you look at a human population, in this room, for example, our DNA is not identical. There are some minor changes, some minor differences in it. And so if we were going to compare, say, your DNA to, say, your DNA, yours might have 3 restriction sites over a particular region, and yours might have only 2, for example, just as an example. So when this particular sample is digested with the restriction enzymes, you would produce 2 fragments, and this one would produce 1. And so it gets its name. If you go through all this, it all makes sense. These are restriction fragments. They're fragments from a restriction enzyme. They are different in length, so restriction fragment length polymorphism. There's several forms of restriction fragments in the population, or now you know why we call it RFLP. So what RFLPs show us is that for any given individual, if you look at 10 or 20 different sites of RFLPs, your particular combination might not exist anywhere else in Iowa City, maybe not anywhere else in Iowa. So RFLPs allow us to identify individuals with different levels of confidence depending on how extensive the analysis is and what population they come from. It also allows us to diagnose genetic diseases. So if one child has a particular condition and the parents want to know whether the next one has it or not, the RFLPs in many cases can pick out the presence of that, or trying to identify whether one parent or the other parent has a genetic trait or whatever. Questions about that? Okay. So this is an example of an RFLP, a restriction fragment length polymorphism analysis. Here is the analysis done on the victim of the crime, and you can see how many different bands are showing up in that one. Here is a blood stain that was left behind at the crime scene. Perhaps the victim defended themselves and the criminal bled a little bit in the location. And for whatever reason, we think that one of these 3 people might have actually been the one to do it. And so when you perform that RFLP analysis on these 3 people, you can see that this individual is matching up right on down the line, whereas the other 2 individuals are very clearly different from suspect 1, very clearly different from the victim themselves. Whether or not a sample, so this blood stain and this suspect are matching. They have the same RFLP pattern. Is that per se proof that suspect 1 was there? That's a slightly different question. That would depend on how likely it is for someone else to have the same RFLP pattern. And how likely it is to have a match or not is going to depend to some extent on the population that the suspect comes from. That there are groups of people that have greater variation and groups of people that have less variation. And obviously in a group that has less variation, the odds of two people having the same RFLP are going to be higher than a group that has more variation. It also raises the question about whether it's ethically justified to sample a whole group. This kind of question started with a case back in England a number of years ago where there was a series of rapes cases that happened, and the local authorities decided to ask every man in the entire county to give a sample of DNA. And in fact, they were able to find the individual who did it. But the question remains, do the authorities have the right to ask that of somebody? Would they have the right to require it of someone? And so on. So it's a very powerful tool, but it's a powerful tool that also raises questions in its own right. I'd like to talk a little bit about I'd like to talk a little bit about model organisms because they've been so powerful for contemporary analysis. And I'd like to point out that the models that we use are good models or bad models depending on the questions that we're asking. And your textbook mentions these four samples as examples of model genetic organisms, and I want to discuss each of them a little bit to give a sense of what kinds of things they're good for and what kinds of things they're lousy for. All right? But they also give you a sense of the genetic variety in the world and the extent to which the complexity of organisms has continued to increase over time. So this first picture over here is a scanning electron micrograph of E. coli, Escherichia coli, which is one of the gut organisms that almost all of us have a copy of. So it's a bacterium. It's a prokaryote. It's small. It grows easily. That's all good. It's not terribly complicated. So you can't study every possible question on E. coli. But if you can study it on E. coli is far and away of all these four organisms the easiest one to grow and to analyze. It has the least DNA. It's almost impossible to kill, and so on. So it's small and not very complex. It is also a prokaryote. Since it's a prokaryote, there are some aspects that only happen in eukaryotes. You couldn't possibly study it in it. So of these three other organisms, the simplest of these three is this. This is a yeast cell, Saccharomyces cerevisiae. You don't need to worry about spelling it. S. cerevisiae is fine. Cerevisiae means beer yeast. The Germans had a lead in the turn of the 20th century in biochemistry because a lot of the biochemicals came from breweries. When you finish making beer, you have a lot of yeast at the bottom. The breweries would sell the yeast to the biochemists, and the biochemists could isolate things to their heart's content. But they are true eukaryotes. They do have many of the features of gene organization and so forth that we have as simple as possible. They have a few introns. They have some development. They have some differentiation. So many of the features that are the difference between prokaryotes and eukaryotes, yeast cells do have. And in that case, you can study that behavior in a yeast cell. These are the bud scars from having given rise to another yeast cell. But obviously, they're single cells. And in this form, you're not going to study anything that involves tissue differentiation, or organization of tissues, or processing information, or immune systems, or any of a large number of other things. So to do that, you need to go to larger, more complex organisms. So the other two organisms are both larger than yeast and more complex than yeast, and they're useful for different reasons. This is C. elegans, Chinorhabditis elegans. It is kind of elegant. I've seen it in the microscope. It kind of looks like, crystal swirling around. It's a worm. I mean, to put a blunt name on it, it's a worm. But it's a worm with very interesting genetics. It has a fixed number of cells. And an English scientist thought 2 decades ago, hey, this might be really good for genetic analysis because we know where everything is. You can look through it as it's growing, so you can follow development by looking through it. So C. elegans has been a powerful genetic model because it does have many of the features that we have. It has specialized muscles. It has a little bitty heart. It has some neurons. It has a hormonal system and so on. But it's a whole lot smaller, grows a lot faster. Even with that, there are some things that it will never do. So anything that involves aspects of the plant world, photosynthesis, plant hormones, starch production, so forth, you would need some kind of a plant system. And this is the plant system that has been developed extensively. It's kind of like the C. elegans of the plant world. It's called Arabidopsis or Arabidopsis thaliana. And it's a little teeny wee mustard plant. If you can see me holding up my fingers, it's about that big when it's full grown. So this is a plant that you can grow from seed to fertile flower in about 2 to 3 weeks. And the fact that it does do many of the things that real plants do, but does it in 2 to 3 weeks, has made it a very powerful system. So how many genes does it actually take to make a human being? And I can see that I actually probably should have revised this table, but we'll get to that. Let's go back down to the low end, to the simple stuff. Here at the bottom, we've got a couple of prokaryotes. Here is Haemophilus influenza. This is one of the bacteria that can be a rather nasty bug if you get sick. It doesn't cause influenza itself, but it can cause secondary infections. Its genome, the size of its DNA, is about 1.8, roughly 2 million base pairs. And it has about 1,700 genes. So you have roughly 1 gene for every 1,000 bases. And it has just the single chromosome. E. coli, again, is the gut bacterium. 2 to 3 times as much DNA, roughly twice as many genes. And it has roughly 1 gene for every 1,400 bases, still a single chromosome. And then if we jump up to a eukaryote, here you can see the yeast cell. This is a eukaryote. It now has 3 times as much DNA, and it has nearly twice as many genes. It's not an order of magnitude different. It's a couple to 3-fold different, right? Its genes are a little bit more spaced apart, but it now has multiple chromosomes. If we keep on going on up this series, we can see that we are getting larger and larger genomes as we go up. Here we are at humans, and we're roughly 3 billion bases. We're roughly 1,000 times, roughly 1 million times as much DNA as E. coli within an order of magnitude. But only 10 times as many genes. In fact, even this number is probably too high. The current estimates for our gene number is now down in the mid-20s. Is that right, Eli? 24 or something? Yeah. So as we've gotten better analysis, the numbers of genes is actually coming down a bit. So now we're looking at a gene density that's only 1 in 100,000 bases. And this is the numerical expression of what I was showing you before, of the idea that when you look at all of our DNA, a lot of it isn't transcribed, and even what is transcribed is not translated. We also see that we now have many more chromosomes involved, and that gives additional layers of organization and regulation as possibilities. Any questions? Okay. So this leads to one of the accomplishments of the recent decade or so, which is that we've gotten to the point where we can actually map the entire genome. The genome of an organism is the whole body of DNA that makes up its chromosomes. So the human genome is roughly 3 billion bases. Genome mapping is made more or less difficult by the characteristics of that organism. So E. coli has very little repetitive DNA. We have a lot of repetitive DNA, and trying to sequence through that was one of the technical challenges that had to be solved. There are some regions that are transcribed but not translated, so that that's an additional feature of the DNA. But having mapped the whole genome, having sequenced it, you now want to identify what those sequences are. Then we need to try to identify where they lie and what they are and so forth. So this is a little piece of a genome map. Each arrow represents a gene. The genes can be found in a number of ways. Some of them are believed to be genes in the sense that they are what we call an open reading frame. That is, they have a start place, they have an end place, they look like they could be, but we don't know. You can identify a gene by an express sequence tag, where you take a bit of RNA that's in messenger RNA, so you know the cell is using it for message, and use that to go back and identify the sequence in the DNA comparable to it. We can also sometimes pick up genes by looking at whether they're similar to other genes. It turns out that the larger the organism, the more we kind of borrow from earlier genes to make a new related but distinct thing. And you can see here different lengths of genes. You can see some are going one direction, some are going the other direction, and so on. This is just showing you that when you then do that, this happens to be for E. coli, you can assign genes to different categories to ask, what is the genome invested in? What does E. coli spend its DNA on in terms of its daily life? And you can see that the biggest chunk for what we know is metabolism, and we'll be talking about that in the middle part of the course. Here we have about 5% that's involved in structure, so that it has a shape and not just, jello. Here's roughly 8.5% involved with macromolecular metabolism, copying DNA, making RNA, translating it into protein. 4% on regulation. It's not enough to make something. You want to make it at the right time. We're going to be focusing on regulation a number of times along the way. 10% on transport. When you think about where it lives, you can see that it's going to want to bring things in and get things out. So that makes some sense, but 10% is still quite substantial. Other activities of various kinds, another 10%. About 2% of the E. coli genome is free riders, organisms that do E. coli no good at all. They're just sort of hanging on because they were there in the past. And then 38% that we don't know what it's for. I find that amazing. We have studied E. coli since the 19th century. And there's 38% of it we still don't know what it's for. So there are definitely frontiers still out there. There's still a lot of life that we don't fully understand. Based on analyses like these of E. coli and others, we now think that probably the minimum number of genes it takes to make a free living cell is somewhere between 250 and 300 genes. This ability to engineer DNA, to rearrange it, to mutate it selectively, to add things and subtract things, means that we can actually engineer organisms. That is, if we use recombinant DNA technology and then get that back into an organism, into its germline, all of its offspring will have that genetic alteration as well. And we call those transgenic organisms. Transgenic organisms are produced for a variety of reasons. This over here is sometimes called golden rice. So this strain of rice has vitamin A precursors. This golden rice can alleviate vitamin A deficiencies if it's used to replace ordinary rice. The plant geneticists who developed it added in somewhere, what was it, 6 genes or 8 genes in order to make it, Eli? Somewhere in that ballpark. So they had to put in a number of steps, but this rice will replace that, and vitamin A deficiency is a leading cause of blindness in the world today. Transgenic organisms can allow us to do research. You think this gene is behind this disease? Well, knock out that gene and we'll find out whether that causes the mouse to have that disease or not. There are transgenic organisms that are producing medicines. Many of the monoclonal antibody-based medicines are actually through transgenic organisms. Obviously, transgenic organisms raise questions ethically, and I don't know that we have time to answer those right now because it's about 4:14 according to my time, but I'd be happy to stay and talk about those if anybody wants to discuss those in more detail. AV person, I think we're done.